Hi everyone, in today's lecture we're talking about protons and bicarbonate, their regulation in our kidneys. So very important for you to know when you're looking at this diagram कि bicarbonate ये ऊपर से आता है तो जितना भी filtrate होता है उसमें bicarbonate भी होता है, right? We already talked about all of these ions. Now we're left with bicarbonate. So bicarbonate either filtrate में आ रहा होता है, this is something that you need to know. और जो protons हैं ये secrete out हो रहे होते हैं हमारे जो proximal convoluted tubule के cells हैं, epithelial cells, उनमें से protons excrete out हो रहे हैं. So again, let's establish that bicarbonate is coming from the filtrate, ऊपर से आ रहा है इधर, और protons secrete out हो रहे हैं. So just know this for now. Moving back, our body it ensures कि हमारा pH 7.4 per maintain. Right? If there are fluctuations in this pH, it can be potentially fatal for the patient. Now, who maintains this pH? तो bicarbonate ions का एक बहुत important role है कि ये ensure करते हैं कि ये pH maintain रहे. Now, what happens अगर for any reason if there are fluctuations in this pH तो acidosis की तरफ जा रहा है patient so what will happen so there will be a lot of protons that will come and that will start binding to this protein तो जब बहुत ज़्यादा protons आकर इस protein के साथ bind होंगे क्या होगा I'll tell you that in a second और दूसरा alkalosis में क्या होता है alkalosis में यही protons जो bound हुए थे इधर so they'll start unduly detaching from the protein so they'll start detaching excessively ठीक है तो यही attachment और detaching binding and detaching का जो सिलसिला है एसिडोसिस में बहुत ज़्यादा बाइंड होने लग जाते हैं प्रोटॉन्स बिकॉज ऑब्वियसली एसिडोसिस में you have like a lot of acid, right? So the acid doesn't know what it has to do. So it's like, you know what, let's just go and combine with the proteins. तो proteins के साथ bind होने लग जाता है. Alkalosis में there's a deficiency of protons. तो ये detach होने लग जाता है. इसी attachment और detachment में the folding of the protein that gets changed and the metabolism is disrupted. So that's the reason why it is very important to maintain the pH at 7.4. अब मसला क्या आता है ह्यूमन बॉडी में that human body is basically an acid producing machine. It constantly keeps producing acids either from the combination of carbon dioxide and water or there's so many other ways. So it's constantly producing acids. अब what happens कि हमारे body में एक बहुत ही there's a very constant reserve of bicarbonate. So that bicarbonate will start feeling threatened कि oh my god there's so much protons then they can neutralize us. So it will start feeling threatened. Now, this is our body's job to ensure that there is sufficient amount of bicarbonate. Otherwise, obviously, it can be fatal. So kidneys ये क्या कहते हैं? They not only produce bicarbonate, but they also conserve bicarbonate. तो ये पूरी back story थी. I'll tell you how they do that. So first and foremost, if you go back to this diagram, you'll see कि bicarbonate यहाँ से आ रहा है, right? और यहाँ से proton secrete हो रहा है. Ideally, I should be starting from here, but let me start from here अभी. तो bicarbonate और protons ये क्या होंगे they'll merge together ठीक है जैसे मैंने आपको बताया कि bicarbonate it is always under threat तो proton आकर इसके साथ लग जाएगा and then our body is an acid producing machine so it will constantly keep producing acid अब ये हमारे पास carbonic acid बन गया this carbonic acid gets broken down into carbon dioxide and water in the presence of carbonic acid अब ये carbon dioxide neutral है compound है so it will easily diffuse through here simple diffusion होगी इधर it'll go up मतलब सेल के अंदर चला गया वहां कार्बन डाइऑक्साइड एंड देयर विल बी abundance of water वाटर जो आया था ना सॉल्वेंट ड्रग में these two things they combine together again in the presence of carbonic anhydrase and form carbonic acid and it will then dissociate into proton and bicarbonate तो हमारा मेन मकसद था ये जो bicarbonate बाहर पड़ा है इसको अंदर लेकर आना without any hindrance without any neutralization तो वो हमने सब कुछ कर लिया हाँ इनिशियली न्यूट्रलाइज हुआ था लेकिन फिर डिसोसिएट भी तो हो गया था एंड इट्स अ गुड थिंग क्योंकि हमें कार्बन डाइऑक्साइड मिल गया जो इजीली डिफ्यूजिबल था ये अंदर आया it got you carbon it got you carbonic acid and then it dissociated into bicarbonate again and then we have our channels here transporters here जो कि bicarbonate को channels basically facilitated diffusion कराकर they'll take bicarbonate into the interstitium and then eventually into the capillaries peritubular capillaries and what about this photon it gets again recycled back it gets sent back इसकी क्या होती है इसकी secondary active transport होती है and then you have this counter transport this is counter transport also known as sodium hydrogen exchanger ये sodium hydrogen exchanger luminal surface पे पड़ा होता है sodium is coming along the concentration gradient down the concentration gradient and proton is being pumped against the concentration gradient और इसकी pumping हो रही है and it is secondary active transport because the energy for this pump is required from this right for pumping hydrogen out or proton out is required from this it's taken up from this gradient और ये अपनी energy ले रहा था sodium ATPase pump से right So eventually proton वापस बाहर आ गया और इसका क्या होगा, proton is continually being recycled. So this is one thing you need to know. और bicarbonate हमने successfully deliver कर दिया अंदर and it went into the interstitium. So there's no rocket science involved here. It's very basic. Let's read this information. Also, many students get confused at this fact. कि अच्छा carbonic acid इधर भी आया, carbonic acid इधर भी आया. So this is the equation: carbon dioxide and water. When they come together, it gives you carbonic acid. So here is where carbonic anhydrase is. This is the enzyme. तो जहाँ carbon dioxide और water आएगा, and it gives you carbonic acid, वहाँ carbonic anhydrase, and also it's a reversible reaction. तो जब आप इसको dissociate कर रहे होते हैं ना, तब भी you'll have carbonic anhydrase. तो आपने बस ये याद करना है कि ये और ये, ठीक है, the product and the reactants. तो यहाँ और यहाँ, और दूसरा again carbon dioxide and water and this. तो इनके बीच में you have to put carbonic anhydrase. Okay, now reading this information from ChatGPT. The proximal convoluted tubule, it does not excrete bicarbonate. Obviously, it does not excrete. इससे तो it has to reclaim all the bicarbonate. जो भी bicarbonate आ रहा है आगे GFR से, it has to take that back into the blood. So 80 to 90% of that bicarbonate is taken, it's reuptaken. Sorry. So this is such a fascinating fact. Other than that, the PCT does what? It secretes protons into the lumen, but this proton is not lost, it is recycled. So see, continual recyclation होती है. So let's look at the step-by-step mechanism. Filtered bicarbonate in the tubular lumen. So first of all, ये जो आपके lumen में था bicarbonate, that was filtered, right? ये तो मैंने already established कर दिया था शुरू में. Bicarbonate is filtered from the blood. It enters the tubular fluid. On the apical luminal surface, this one, you have sodium hydrogen exchanger that secretes protons and driven by sodium reabsorption. So sodium is reabsorbed and protons are pumped out or secreted out. In the lumen, what happens is that the proton that went out and bicarbonate from the filtrate, they come together and they form carbonic acid. And this carbonic acid breaks down in the presence of carbonic anhydrase and it forms carbon dioxide and water. Now this carbon dioxide, it can easily diffuse, है ना? अब ये जो carbon dioxide बना था, this can easily diffuse inside. And once it goes in the tubular cell, what happens is that it again forms your carbonic acid and then it again dissociates into bicarbonate and proton. So this bicarbonate is reabsorbed into the blood via sodium bicarbonate co-transporter. अब यहाँ हमारे पास basolateral surface में sodium bicarbonate co-transporter है. तो यहाँ you also have cells, you have मैंने यहाँ transporter बोला था and then I also said channels, but you have very important sodium एंड बाइकार्बोनेट को ट्रांसपोर्टर, तो दोनों एक ही डायरेक्शन में जा रहे हैं. सो सोडियम और बाइकार्बोनेट को ट्रांसपोर्टर आपके बेसोलेटरल सरफेस पे पड़ा है जो कि इनको रीअब्सॉर्ब करता है बैक इनटू द ब्लड एंड प्रोटॉन इज सिक्रेटेड बैक अगेन टू रिपीट द साइकिल. So proton is continually being recycled whereas bicarbonate is effectively reclaimed. So again, a summary. Your proximal convoluted tubule, it reabsorbs 80 to 90% of the filtered bicarbonate and the enzyme is this, and you have sodium proton exchanger on the luminal surface and you have your sodium bicarbonate co-transporter on the basolateral surface. So summary क्या है कि proximal convoluted tubule has to reclaim the bicarbonate और जो hydrogen है, it is continually being recycled. So that's pretty much it.